previous video, we started to jump into genetics and inheritance. We looked at Mendel's laws. We saw that Mendel was the father of genetics. We looked at some important concepts that you need to know for genetics and inheritance, such as what is an allele, dominant and recessive alleles, phenotype, genotype, homozygous, heterozygous, and then, yes, the law of dominance and the law of segregation. Then, if you don't understand, if you haven't yet comprehended what these terms mean, don't worry. By the end of this video, you should have a better understanding of those terms once we have done monohybrid crosses. So, in today's video, we're going to look at the format for representing a genetics cross. We're going to look at the types of dominance that you get, and then we also are going to do genetics problems involving each of these three types of dominance, and then we're going to look at the proportion and ratio of genotypes and phenotypes. So what I'm quickly going to do is just a recap of some of the terms that we did yesterday, or some of the work that we did yesterday, just before we go into these crosses. So if you remember, we said that the dominant trait is always represented by a capital letter, and it is written first when you're out of the dominant and the recessive trait. The recessive trait is written with a lowercase letter, and then it is written after the dominant trait. So using Mendel's pea plants again, tall and short plants, a tall, that's shorts, tall and short plants. So a tall plant is the dominant one over the recessive short plant. So when you do write the genotypes, you take the first letter of the dominant trait, and in this case it would be T for tall, and the recessive trait would then be a lowercase t. Then you would also remember that yesterday we did the term homozygous and the term heterozygous. So when something is homozygous, it is the same. So if we talk about a homozygous tall plant, we're saying that both the traits are tall, or both the alleles. That is a correct term to use because these represent alleles on a chromosome. So the traits. So homozygous tall would look like that, and then homozygous short would look like that. And this is the only time that a short, where you will find a short plant to see it, is if it has homozygous short, because the recessive trait will only come out if there are two of it. Then heterozygous means different, so a heterozygous tall plant, because remember, even though we do have the short allele there, the dominant allele, which is tall, overshadows it, so what you will see in the phenotype is in fact tall. So those things are very important when we go into these crosses. Before we go into those crosses, we need to look at the types of dominance that occurs. Complete dominance is basically what we've been looking at the whole time. It is when one allele is dominant and the other is recessive, such that the effect of the recessive allele is masked by the dominant allele in the heterozygous condition. Then incomplete dominance, none of the two alleles of a gene is dominant over the other, resulting in an intermediate phenotype in the heterozygous condition. So in this case, basically, you're creating something new. But we'll look at that when we do that cross. Codominance, both alleles of a gene are equally dominant, whereby both alleles express themselves in the phenotype of the heterozygous condition. So let's delve into these monohybrid crosses. And a monohybrid cross, what is that? It is a cross where only one characteristic is looked at. Now, in the word mono, as we all know, means one. You guys should know this by now. Mono means one. So an example here, a tall or short plants, wrinkled or smooth seeds, yellow or green seeds, once again using Mendel's pea plants because they're very easy to use as an example and they're also very straightforward. So in a monohybrid cross, you only look at one of these characteristics or traits at a time. So you'll either be looking at the tall or the short, the length of the plant, you'll be looking at whether the seeds are wrinkled or smooth, or you'll be looking at the color of the seeds. So only one of these characteristics is looked at once. Now, complete dominance examples. So this is actually a question from an old exam paper. I'm going to do this with you because when we do monohybrid crosses or any crosses for that matter, there is a format that you need to use in order to answer these questions. So I'm going to do this with you so you can see the format and you're going to understand it better than me just talking and rambling on about it. Now, in pea plants, the allele for a tall plant, so they're telling us that there's a tall plant, is dominant. So they're telling you that the tall plant trait is dominant and they're giving you the allele basically, and they're telling you it's a capital letter T. To the allele for a short plant, so that one is definitely recessive because it is a lowercase. A homozygous tall plant is crossed with a homozygous short plant. Now this is very important because this is going to tell you how you're going to start your cross out with. So if something is homozygous tall, once again it's going to look like that. Crossing with a homozygous short, it will look like that. Use a genetic cross to determine the offspring. Okay, so the first thing that you do is you write P1. Now P stands for parental, and then when we finally get to the F generation, F stands for filial. Now filial is just a fancy word to use in order to describe the offspring. So P1, why do we write P1? Because this is the first parent generation that is being crossed. You can get a P2, which we'll look at later. That is generally when you use your offspring as parents and then they start crossing. But before you can get there, you need to do the P1 first. So P1, then you write phenotype. So phenotype, remember features. What do you see when you look at these plants? So you can write homozygous, homozygous tall, crossing with homozygous short. Running out of space there. So that is your phenotype. Very easy. Then genotype. I just want to emphasize that you guys do remember to write P1. That is very important. Don't just write the P. You need to write P1. Genotype. So this is now the alleles that we are talking about. So if it's homozygous tall, it's going to look like that. Crossing with homozygous short. That looks like that. Then, what has to happen here if these parents want fertilization to take place? They have to go through the process of meiosis. What is meiosis going to do? It's going to give us gametes. So basically what the gametes are going to do is we're just going to separate those alleles from each other. So you need to write it separated like this. If you don't put a comma, just make sure you write them very far apart. I've even seen in when I was marking papers that kids tend to put them in boxes like that. That is fine, but that's a big hassle. You can just write it like that. Okay, so now we've got the gametes. What has to happen now? Now fertilization needs to occur. And there is two ways of doing this. I prefer using a Punnett square. So this thing is what we would call a Punnett square. I'm just using a table that I created. You are going to draw this with a ruler so it's nice and neat, so that the person marking your papers doesn't have to cringe when they're marking it. So now what you're going to do is you're going to take the gametes that you have here and you're going to place them in these boxes for crossing. So the first parent obviously is the homozygous tall, so you're splitting the two alleles. Then the other is the homozygous short, and it's going to look like that. Now, how do you cross it? Basically, this is like a little game. So this column going down, this one is also going down, and then that one is going to the side. So if we look at this T, you can write it down here. Remember, the tall plant is the dominant one, so the dominant characteristic is always written first. So T going down, this one will also be a T going down. See it going down in that column. And now you're going to do the homozygous short, so it's going that way crossing. So there will be a T and there will be a T. For this one, same as well. Do you see how easy it is with this cross? There is another method, but this one is, I find, the most structured, and it will also, well, you won't, the chances of you making mistakes here is very slim. Okay, so now we've done the fertilization part. Now we need to show what these offspring look like, because this is all the possibilities of the offspring that could be produced when these two parents cross. So these are all the possibilities, and in this case they all are the same. So let's write the F1 generation. So now we are, that's bad spelling. So the F1 generation, and we are now going to start with the genotype. Just, you don't need to write that, I just wrote it for explanation. So you can just write F1 genotype. So what do we have here? We have a genotype that looks like that. Or you could just write 4 T lowercase and a capital letter, because how many are there? 1, 2, 3, 4 of them. Okay, do you understand? I hope you do. Then phenotype. So the F1 phenotype, what is that? It is the features. What do these represent? So capital letter T, lowercase t, that would be heterozygous tall. Okay, heterozygous because they are different alleles. The dominant gene is carrying around this little recessive gene over there. And then sometimes they can ask you to put it in ratios. So in a ratio here it won't really work, but in this case you can write it as a percentage, and you can say 100% tall. And if you want heterozygous tall. So there is the first genetic cross, the first monohybrid cross, and it's really straightforward. I'll do another one with you now. So let's quickly look at the marking allocations. Where do you get marks for these genetic crosses? They generally count out of 6 marks, and this is a memo like the ones we've been doing in class. That is the other option, this one over here, but I don't like that one because it can get confusing. So what do you get marks for? So firstly, any 5 marks plus the 1 compulsory mark. So in this case, you had to give a percentage of the offspring. What were they? Heterozygous or homozygous? What? Then parents and offspring. So you get a mark for writing P1 and F1. So you would get a mark for these two. When I marked papers, I drew like a connecting line like that to show a mark. Then you also get a mark for meiosis and fertilization, also put a mark like that. If you wrote meiosis and fertilization, you would have already gotten a mark. Then you would have gotten a mark if your phenotype was correct in the P1 generation as well as the genotype. You would have gotten a mark for gametes, and then for the F1 generation, if your genotype was correct as well as your phenotype. So see the numbers that they put there, we'll do one like that now. So this is where the marking is allocated when we look at these genetics crosses. Now going back to this, what I want to do is I just want to do another example. So let's say, for example, they have given you a heterozygous tall plant crossing with a homozygous short plant. So we can say that this is the P1 phenotype. And then the genotype of this one would look as follows. So heterozygous tall, it would mean that the alleles look like that. Homozygous short would look like that. Then we go through the process of meiosis, and that gives us our gametes. Remember to separate them. Okay, and then you would go into fertilization. Remember to write fertilization. As you saw that you would get a mark for that. Now, let's quickly put in these alleles for the gametes. Okay, remember this one goes all the way down, this one goes all the way down. So if it's dominant, it is written first, and this one is recessive, and then this column goes that way. So that's what it will be looking, and then there is a group of recessives together. Then why is it not writing? F1 generation, you don't need to write that. Let's do that over, it's bothering me. F1 genotype, it's easier writing the genotype because it just helps you pull everything out of these blocks so that you know what to write when you get your phenotype. So in this case you can write 2 and 2. Why? Because there's 1, 2 of those, and there's 1, 2 of those. So phenotype would then be 2 heterozygous tall and 2 homozygous short. So if you want to put it percentage-wise, you could say 50% heterozygous tall and 50% homozygous short. If they ask you for ratios, you could say it is 2 to 2, or simplified 1 to 1. So there's another monohybrid cross. It's quite easy. I think I've gotten an example or a picture one here at the top. So this is for brown eyes and blue eyes. Obviously it's not the whole format, it's just showing the Punnett square. So if somebody has brown eyes is the dominant allele, and then the recessive allele is then blue eyes. So in this case, this person had heterozygous brown eyes, crossing with somebody that had heterozygous brown eyes. So going down to the side, to the side. So BB it would have been written, and then lowercase b there. So in here it would have been homozygous brown, heterozygous brown, heterozygous brown, and homozygous blue, because there are 2 recessive alleles that have joined there. So this is the possibilities of the offspring. If you had to put that, the genotypes, how you would have written that would be 1BB, 2 and 1. So phenotype would have been 1 homozygous brown. I'm not going to write out the full thing. 2 heterozygous brown, 1 homozygous blue. Okay. In this case, they used a homozygous blue, so the recessive gene, crossing with a heterozygous, and then that is the result. I'm not going to go through that now again. I'm sure that you understand it. So when they ask you for a P2 generation, they will clearly state that in the paper. So let's quickly use this as an example. So if we have to do a P2 generation, the P2 generation is technically the F1 generation. So let's say we are crossing 2 of these offspring with each other. So where did I write that? Okay, let's start over. So P2 is when you use the offspring to cross with each other. So the P2 phenotype would be, phenotype. So it would be heterozygous tall crossing with another heterozygous tall. Then the genotype, it would look like that. Then we go into meiosis, giving us gametes, and then the process of fertilization occurs. So can you see how I'm splitting them? Remember that one goes down to the sides, so this T is going all the way down, this T is going, because it is recessive, let's just put it somewhere more at the back. Then this T has to go all the way through. Okay, so already it's looking interesting. So if we now need to write, remember, it is F2 because we are busy with P2. So obviously the second generation will have second generation offspring. So F2 genotype, it will look like this. And then the phenotype will be, so 1 homozygous tall, 2 heterozygous tall, and 1 homozygous short. Okay, so there we go. I'm going to end this video here so it's not too long for you, and then I will do, and the next video will be on incomplete and codominance.